Hello all. Thank you so much for joining. As you're probably aware, we will be presenting a webinar today. Our own Charlene Vlichko from DiscoverX will be presenting new tricks for old drugs, an unbiased phenotypic approach to drug repurposing. Charlene is our Director of Research Biology for the Phenotypic Drug Discovery Services branch of DiscoverX. Has been with the BioSeq/BioMAP group for a number of years and pretty much as much an expert on the technology as they come. So I just wanted to go over a few clerical notes. The slides will actually be available for download and viewing after the webinar, so a link should be going out shortly after, so anyone who has to leave early or wasn't able to watch this should have access. Secondly, if you have any questions at all, feel free to ask. There's a little Q&A box at the bottom right. Just pop them in there, and we will queue them up and have Charlene answer all your questions at the end of the presentation. So without any further ado, I give you Charlene, let's go.
Hi, Adam. Thanks for the introduction. So, as Adam mentioned, the topic of my webinar today is New Tricks for Old Drugs, an Unbiased Phenotypic Approach to Drug Repurposing. There we go. All right. So, drug development in today's market is very challenging, and the average cost to bring a drug to market is now estimated to be well over $1 billion and take anywhere from 10 to 20 years. And a staggering number of compounds are failing along the way, with an estimated 20% of those that even make it to phase 2 ultimately gaining final approval. So there's a growing amount of interest and activity by drug companies in drug repurposing, also known as drug repositioning or rescue. And there's ample reasoning behind this. Repurposing can salvage compounds that have failed clinical trials for their primary indications. This can also extend patent life on existing approved therapies, thus avoiding the patent cliff expirations that cause large decreases in sales due to competition from generics and biosimilars. And finally, the approval of repurposed drugs is often accelerated due to pre-existing clinical safety data. So because of this, the cost of getting a repurposed drug approved for a secondary indication can be a fraction of the cost of initial drug approval. But that isn't to say that repurposing programs are without their own set of challenges. One of the largest challenges is that repurposing is still a relatively new area for many companies, and as such, there is no standard pipeline that currently exists. Many of the current success stories have been serendipitous discoveries, such as sildenafil and bimatoprost, but a more integrated approach can identify repurposing indications earlier on, possibly before approval for the primary indication. There's a need for evidence-based guidance on indication selection, ideally a systems biology approach that integrates knowledge gleaned from multiple data sources, can help guide indication selection, and also identify response biomarkers early on. One repurposing approach that's gaining traction is the use of combination treatments, which may have increased efficacy in complex diseases where selective polypharmacology may be advantageous. However, a systematic evaluation of potential combination partners is needed for prioritizing the exponential number of combination possibilities. BioMAP is a phenotypic screening platform that consists of human primary cell cultures that are stimulated in a variety of ways in order to model different disease and tissue types. Compound effects on a number of clinically relevant biomarkers are measured and displayed as a BioMAP profile, which visualizes both effective biomarkers and dose response across multiple systems. In addition, we've built a large reference database of BioMAP profiles generated from over 4,500 compounds, including drugs, failed drugs, tool compounds, and includes small molecules and biologics. And we've developed proprietary tools to mine this rich data source. As you know, multiple cellular processes play a role in any one disease, which is why approved treatments can represent various target classes. Likewise, there may be additional molecular drivers of disease that have not yet been identified. Target-agnostic phenotypic screening offers the advantage of modeling the complex drivers of disease while also allowing for the testing of various hypotheses. The BioMAP platform incorporates a suite of tools that can be used to address the needs of drug repurposing programs. The main tools are our panels. So we've grouped our BioMAP systems into various standard panels. These panels can cover broad biology, allowing for the simultaneous evaluation of compound effects on diverse tissue and disease biology. We've also developed more disease-focused panels for fibrosis, oncology, and autoimmune disease. As I previously mentioned, over the past 15 years, we've built a large reference database of BioMAP profiles. Because these compounds include approved standard of care drugs and failed compounds that represent many different target mechanisms, we have been able to develop a rich knowledge base and a suite of informatic tools to interrogate this data. An example of this are the signatures of safety and toxicity that we've been able to develop based on BioMAP profiles of compounds that have failed clinical development due to adverse effects. And finally, we've developed a service that allows you to evaluate the phenotypic effects of two compounds in combination at multiple concentrations and compare them statistically to single agent treatment alone. This is useful to companies that are evaluating combination therapy as a way to repurpose their existing drugs. So these tools can be combined in a variety of ways to fit the needs of a particular drug repurposing program. So for example, profiling in the Broad Diversity Plus panel may yield additional mechanistic insight of your drug on unexpected tissue types or disease states. And an unbiased similarity search of our reference database may yield information in regards to additional targets or potential mechanisms. These hypotheses can then be tested in one or more of our disease-focused panels, which allows for the identification of disease-specific biomarkers. So the first case study I'm going to present today is an example of this approach. Dasatinib is an approved treatment for CML, which I will show you may have very good potential for the treatment of fibrotic disease. Dasatinib, which is marketed commercially as Sprycel, is a BCR-ABL inhibitor that has been approved for the treatment of Philadelphia chromosome-positive chronic myelogenous leukemia, as well as acute lymphocytic leukemia. It has known polypharmacology, so its other targets include SRC, c-KIT, Eprin, PDGFR, and GFR, and it is also not the only approved BCR-ABL inhibitor in the CML treatment space. Other approved BCR-ABL inhibitors include Imatinib, Erlotinib, and Ibrutinib. Patents for Dasatinib will begin to expire in about 4 years, so the question is there opportunity to expand Dasatinib's market reach by expanding to new indications? It is challenging to determine where to start looking for new indications given the breadth of possibilities. At the same time, you don't want to look too narrowly and miss potential opportunities. So is there an evidence-based approach that can be used to identify new indications for already approved drugs such as Dasatinib? The BioMAP approach allows for the evaluation of Dasatinib in diverse disease settings to identify novel activities. Further evaluation in a disease-focused panel allows for the testing of certain hypotheses, generating evidence to support additional indications. So this is a profile of Dasatinib in our Diversity Plus panel, which covers broad biology. Just to orient you, Dasatinib was tested in 12 systems representing various tissue and disease biology. These systems are denoted by the circular icons at the top, and each system is associated with specific readouts which are listed along the y-axis that are clinically and biologically relevant to the diseases that they are modeling. Dasatinib was tested at 4 different concentrations, and the effect on biomarkers is normalized to vehicle control, and the effects of all 4 concentrations are displayed together on a single plot to evaluate dose response. So the first thing that we notice is that the anti-proliferative effects, which are denoted by these gray arrows, were observed for multiple cell types. This is something that is characteristic for compounds developed for oncology applications, but it may not be a desirable trait for development for autoimmune indications. Strong anti-inflammatory effects on systems modeling T and B cell activation are observed, consistent with its role in CML. And interestingly, Dasatinib has a lot of activity in these systems, which model tissue remodeling and fibrosis, including effects on matrix-related molecules such as collagen and MMPs. This leads to the hypothesis that Dasatinib may be useful in treating fibrotic diseases. So when we performed an unsupervised similarity search of our database with Dasatinib, Nintedanib shows up as one of the top similarity search matches. Here we have an overlay of those profiles with Nintedanib in black and Dasatinib in pink. Nintedanib is a multi-angiokinase inhibitor that is FDA approved for the treatment of idiopathic pulmonary fibrosis, otherwise known as IPF. This similarity of Dasatinib to a clinically approved drug for fibrosis strengthens the case of Dasatinib's efficacy in fibrotic disease. So we've gone from broad profiling of Dasatinib and used the analytical tools and database to identify a close similarity search to Nintedanib. We then profiled Dasatinib in our focused BioMAP Fibrosis panel. This panel consists of 3 BioMAP systems that were constructed to model myofibroblast activation and the interplay between myofibroblasts and epithelial cells in various tissue contexts. So this is a fibrosis panel profile of Dasatinib. We can see here that Dasatinib is very active in this context and modulates a large number of biomarkers. So what does this all mean? So we're going to break it down. First, we see that Dasatinib decreases the expression of sentinel markers of myofibroblast activation in all 3 systems, alpha-smooth muscle actin and cadherin. Myofibroblasts are considered to be the key pathogenic cellular player in the development of fibrotic disease. The production of collagens by myofibroblasts are also decreased with Dasatinib treatment. Excess collagen production leads to the distortion of tissue architecture, and in the lung, this leads to the loss in lung function. And finally, we see a decrease in the expression of pro-inflammatory cytokines such as IL-6 and IL-8. The expression of both of these cytokines have been reported to be upregulated in patients with IPF. And interestingly, we actually see an upregulation of IP10, otherwise known as CXCL10, in the system that models pulmonary fibrosis. IP10 is considered to be an antifibrotic chemokine, and upregulation of this molecule may be beneficial for treating fibrotic disease. So in conclusion, broad profiling of Dasatinib in the Diversity Plus panel identified both antifibrotic and immunomodulatory activities. A similarity search against a reference database revealed many shared activities with Nintedanib, an already approved treatment for IPF. And then finally, profiling in the focused fibrosis panel characterized additional antifibrotic activities of Dasatinib and identified potential biomarkers that are relevant to disease. So in terms of repurposing, this data helps support the therapeutic expansion of Dasatinib to fibrotic diseases such as IPF. Localized delivery of Dasatinib to lung tissue could potentially avoid any side effects that might be associated with long-term systemic administration. And repositioning Dasatinib may allow accelerated entry into a relatively uncrowded market, which is estimated to exceed $1 billion by 2017. So I'm now going to present another case study surrounding Ibrutinib, a BTK inhibitor with known polypharmacology. The question is, does this polypharmacology lead to expanded indication potential? Ibrutinib, which is marketed as Imbruvica, is a first-in-class Bruton tyrosine kinase, or BTK inhibitor. It has been approved for several different blood cancers, and kinome scan analysis, shown here, reveals that Ibrutinib inhibits additional kinases with an IC50 value close to that for BTK. Many companies are developing second-generation BTK inhibitors with greater selectivity, so Ibrutinib is at risk of being replaced by these compounds as they gain FDA approval. The question is, what are the phenotypic effects of Ibrutinib's polypharmacology? Are these activities on other targets besides BTK a liability or an asset? The BioMAP approach that we took here was similar to Dasatinib in that we utilized broad profiling to characterize these additional activities and expand to possible new indications, and then we deployed our proprietary analytical tools to identify potential mechanisms and liabilities. This is a Diversity Plus profile of Ibrutinib. Activities in the BT system modeling B cell activation are consistent with BTK inhibition. However, direct inhibitory activities on bronchial epithelial cells is not consistent with BTK, and neither is a direct effect on matrix-relating proteins. So the question is, are these due to BTK inhibition or to some of the other targets of Ibrutinib? So we then compared the profile of Ibrutinib to two newer BTK inhibitors in our database, GDC0834 and RN486. Compared to these newer BTK inhibitors, Ibrutinib is more active in non-immune cell-based BioMAP systems, which are denoted by these black brackets. I've annotated the activities that are unique to Ibrutinib. These may be due to Ibrutinib's other targets. So the question is, can we associate these activities with a known target or mechanism class? So we've developed consensus profiles for a number of distinct target and mechanistic classes. Because we have a number of individual agents in our reference database that share the same target class, we have merged these profiles into consensus profiles. To compare these various profiles to compound of interest, we have displayed these activities as a heat map with increased activities in red and decreased activities in blue, and the individual biomarkers and systems are listed on the right-hand side of the heat map, with the classes and the compounds listed on the bottom. So I'm zooming in on several of the non-immune cell-containing systems that we see activity for Ibrutinib. And what we can observe is that many of these activities are shared with the EGFR inhibitor mechanism class, and I've highlighted them in yellow here. So now I'm overlaying the profile of Ibrutinib with Erlotinib, a specific EGFR inhibitor approved for the treatment of NSCLC and pancreatic cancer. This confirms shared activity in these non-immune cell-containing systems. One of these shared activities is the increase in EGFR expression in the HDF3-CGS system, which models dermal wound healing and inflammation. So skin toxicity is actually a commonly reported adverse effect of patients treated with Erlotinib, as well as other EGFR-I therapies. In fact, the presence and severity of skin rash is associated with Erlotinib efficacy in cancer patients. And interestingly, skin rash is also a commonly reported adverse effect associated with Ibrutinib treatment and may be due to its activity on EGFR biology. So broad profiling of Ibrutinib in the Diversity Plus panel in comparison to other BTK inhibitors allows for the characterization of the phenotypic impact of both BTK and non-BTK activities. Our analytical tools identified EGFR inhibition as a mechanism associated with the non-BTK activities of Ibrutinib, and in fact, it's now been confirmed and published that Ibrutinib has direct effects on a number of EGFR mutant cancer cell lines, and Ibrutinib is now being clinically investigated as a solid tumor therapy. And skin rash may also be a predictive marker of clinical efficacy in this context as well. So the BioMAP platform can provide a standardized approach for prioritizing both drugs and indications in the repurposing space. We provide analytical tools that can inform our mechanisms and therefore therapeutic indications. Our disease-focused panels can identify clinically relevant biomarkers for particular disease indications. And finally, although I didn't have a chance to talk about this today, we do provide a combo-like profiling service, which allows for the target-agnostic evaluation of drug combinations for both efficacy and safety. And so this brings me to the end of the presentation, and I thank you for your attention.
Thank you so much, Charlie. That was really great, and yeah, now we'd like to open it up to any questions. If anybody would like to make some, all you have to do is click on the little Q&A box in your bottom right corner, turn it on over, and Charlie should be able to answer. So if one come in, how do you think you would follow up on the Dasatinib study you presented?
So that's a really great question. I think that BioMAP profiling gives significant in vitro evidence to support pursuing fibrosis as an indication. And the next step would obviously be to potentially evaluate Dasatinib in some of the animal models for fibrosis, so either the CKD model for kidney disease or the bleomycin mouse model for pulmonary fibrosis.
Right. We have another one here. Have you tested other BCL-ABL inhibitors in focus panels of fibrosis? See how they compare to determine the fact not due to BCL-ABL inhibitors?
Yes, so we've actually evaluated, I didn't get a chance to show this data today, but we've actually evaluated imatinib in the BioMAP fibrosis panel, and we confirmed that imatinib has almost no activities in the fibrosis panel, so the efficacy that we see for Dasatinib is likely due to its non-BCR-ABL activity.
Very interesting. I have another one for you. Are there examples of successful repurposing outside of kinase inhibitor field?
Sorry, could you repeat that one more time?
Of course. Are there examples of successful repurposing outside of kinase inhibitor field?
Oh, yeah, absolutely. I mean, so I think I mentioned previously, but one of the best examples is probably sildenafil, which is, I believe, a PDE inhibitor. It was originally developed for hypertension, but obviously is now used for erectile dysfunction. And bimatoprost is, I believe, a prostaglandin drug that was originally developed for glaucoma and is now used to lengthen eyelashes. So there are definitely multiple drug classes in which repurposing has been shown to be advantageous.
Would you miss biology or miss similarities between two drugs if they only shared activity in one or two of your models, but not all of them?
So I think it's interesting, and that's a very interesting question. I think one of the advantages to BioMAP profiling is that we give you an overall picture. So we do provide analytical tools which will allow you to score overall similarity between two different drugs. But at the same time, we allow you to look at the similarities on a number of different biomarkers. So in the case of Erlotinib versus Ibrutinib, overall, you could see that there were some pretty significant differences in their activities, but it was the activities that were similar in a specific subset of systems or specific biomarkers that are very sentinel to EGFR-I therapies that really led us to the conclusion that one of Ibrutinib's mechanisms of action may be due to inhibition of EGFR activity.
Perfect. And so the last question is, how long does it take to screen with BioMAP, and how much compound would you need?
Yeah, that's a good question. So I think it depends on the concentration that you want to test your compound at. So, and typically we ask, because we're testing 12 different systems, we do ask for a certain amount, and if you're interested, you could always get in contact with your account manager. But because we run this in a standardized methodology, it really allows us for a fairly quick turnaround time. So our turnaround time is definitely in the order of weeks versus months is what it would take for an animal model. So definitely you get a quick turnaround time to test your hypotheses.
Well, thank you so much, Charlene. And I just want want to reiterate that everybody in attendance should be receiving an email in the near future to get a link to be able to view this webinar at your leisure. So with that, thank you all so much for attending, and we hope to see you again soon. Take care.